Good morning. I'm Dr. Sumant Kumar, professor in the Department of Biochemistry. Today I'm going to take the class on polymerase chain reaction topic. So now going to this polymerase chain reaction, before that we should know what are the contents that I'm going to cover in this particular class. So here today we are going to see the principle of the polymerase chain reaction, the procedure, the entire procedure, and what are the advantages and disadvantages of this particular PCR. Now, what is PCR? This PCR is just like taking the Xerox copies. So, it is a technique or a procedure where we will have a particular DNA of our own interest, and that DNA will be amplified and will be taking number of that particular DNA samples. So, without the use of a cell, instead of taking a cell, it is a cell-free amplification technique where our specified DNA which is of our interest that can be generated in large quantities. So that is the polymerase chain reaction. So now in this polymerase chain reaction, so the thing what we have to observe is, so the one is target DNA. So that is the DNA of our interest which has to be amplified. The second thing is pair of primers, where when we are taking the parent strand, from that we have to develop the daughter strand. For that we require the primers which will be annealed to that particular mother strands, that is the parent strand, and along with that one we require the DNTPs, that is deoxynucleotide triphosphates. And the main important one is the enzyme, that is thermostable DNA polymerase, which is that enzyme which is required for the catalyzing this particular reaction. And another one is magnesium, which acts as a cofactor for this particular enzyme. And one more very important one is the buffer solution, which will be maintaining the pH and ionic strength, so thereby this particular enzyme can be active in that particular medium, and so thereby it can do its polymerization reaction. Now actually when we are talking about the polymerase chain reaction, we will be having three main steps. Now what I kept here is primary step in the PCR. This doesn't come under those three steps, but the thing is, this is the foremost part what we have to do when we are going to start the PCR. So here what the thing is, we will be collecting the sample and we will be isolating the DNA. So this is the primary step in the polymerase chain reaction, and the second one is preparing the primers. So we require two primers, because when we are separating the parent strand, we require two primers for each parent strand. So these primers are the oligonucleotides of around 15 to 25 or 15 to 30 nucleotides length, which should be complementary to the regions of the target DNA, that is the mother strand. So in this way, where you can see here, so here you can see this particular strands, the mother strand will be there, and this particular mother strand will be annealed with the help of these primers, which are the oligonucleotides. Now coming to the enzyme DNA polymerase, specifically here the DNA polymerase will be Taq polymerase, which is called as Thermus aquaticus, which is a thermophilic bacterium, and it should be heat resistant. Why specifically we will be taking this Taq polymerase is, we will be taking different types of temperatures going up to 90 and 95 degrees centigrade. So at that point of temperature, this particular enzyme should not get inactivated or should not become non-functioning. So thereby the heat resistant enzyme should be selected. For that particular purpose, we will be selecting or we will be taking this Taq polymerase, which acts as a DNA polymerase enzyme. Now coming to the next one. Now this is the main thing, this is the actual technique. That is what I said previously, that there will be three steps in the polymerase chain reaction. So the three steps in the polymerase chain reaction are, one is denaturation, which is also called as melting or separating the parent strands. The second one is renaturation or annealing, that is the pairs will be getting the primers will be getting paired with the parent strand. The next one is synthesis or extension of these particular primers. And this is the instrument where the total procedure will be taking place. This is the apparatus to perform about 35 cycles, and temperature procedure. So the temperature should be maintained in this manner. The first step where we are doing the denaturation of DNA, at that point the temperature should be around 90 to 95 degrees centigrade. Later, when we are doing the annealing with the primers, at that time the temperature should come in between, somewhere between 15 to 60 degrees centigrade. Later, when we are doing the extension of the primers, again the temperature has to be increased to 72 degrees centigrade. Now coming to the first step, where the denaturation or suppression of the mother strand or the parent strands will be taking place, where the temperature should be maintained at 95 degrees centigrade. At that point of temperature, DNA will get denatured and the strands will get separated. The second thing is annealing, that is or else it is also called as renaturation, where the primers will get paired with the mother strand. So here the temperature from 95 degrees centigrade should be brought to 55 degrees centigrade, where at that point only the annealing will take place. So here the primers will pair with the complementary regions of the target DNA. So here like this you can see the two strands, the black colored strands which are the parent strands, and you can see the primers, one is in orange color and another one is in blue color. So they will be attached to the 3' end of the parent strand. Now they will be getting extension from the 5' end to 3' end. So primers will bind to the complementary sequence of the target DNA. Now in the third step, where the primer extension will be there, in this manner, where the blue color, small blue color arrow mark is getting extended in the form of a large blue color arrow mark, you can see. So in this direction, that is the new strand will be extended or get synthesized in the direction of 5' to 3' direction. Now after completion of this particular process, in the same manner, these cycles, what I said, the 3 cycles, denaturation and renaturation, against the extension, these 3 steps, these procedures will be continued like cycles, and at the end we will be having empty number of DNA strands of our interest. So this after PCR, when that has to be visualized as the results, and the results will be visualized in this manner. So after the completion of the total gel run in the electrophoresis, DNA bands will be visualized after staining them. So this is the overall picture of the procedure what we will be doing. That is the instrument. So where the PCR technique will be taking place in that particular instrument. So in that, this agarose gel electrophoresis will be done, and after the gel electrophoresis is completed and after staining and everything, at the end we will be visualizing the strands, and finally we will be getting the product that will be seen in this particular instrument. Okay, now this is the overall picture or the procedure of PCR, where number of cycles will be taking place. So at the end we will be getting the number of DNA strands. Okay, so that's why the one of the advantages of PCR is it is a very quick one. So quick, and it is a cell-free amplification technique, where instead of getting a single copy we require most number of time. Here with this particular technique we can get some thousands of the same DNA copies within a small period of time. So that is one advantage of this PCR, and it is reliable, both it is sensitive and also specific procedure. Now coming to the disadvantages, we require a particular equipment, and these particular equipments are required for the process to take place. And the stack polymerase enzyme is expensive, so in the small laboratories we can't perform this PCR technique. It requires a particular sophisticated laboratory is required, and rarely if there is any contamination in the procedure what we are doing or the medium what we are taking, so at that time there are chances of getting false reactions. This can be minimized by taking care when we are performing, before performing the PCR, by taking care that everything is perfect and normal. Now these are the different types of PCR or the variants of PCR, like long PCR, that is used to amplify the DNA over the entire length up to 25 kilobytes of genomic DNA segments clone, and nested PCR, where it requires 2 consecutive PCR reactions will be taking place, where the first PCR will get completed, and after that the product of the first PCR will be taken into the second PCR technique. The next one is inverse PCR, where it is used to amplify the DNA of unknown sequence that is just adjacent to the known DNA sequence. Quantitative PCR, which will be compared with the standard DNA. And hot start PCR, it is used to optimize the yield of the desired amplified PCR technique, and simultaneously it is used to suppress the non-specific amplification. So these are the different types of PCR. So on this PCR is nowadays it is being highly used in a number of laboratories to get the result within a small period of time. So thereby it is used to diagnose number of disorders and diseases within a small period of time. So this is about the PCR technique. Thank you very much.